We'll just give it here a minute and then we'll be underway.
You guys are ready to roll.
Okay, great. Thank you, Robin, for the assistance with the Zoom webinar, and welcome to all of our attendees and panelists to today's conversation about vaccines. With the recent announcement that vaccines are now widely available for any Montanan ages 16 and over, we know that there are some questions that people might have about vaccines, how vaccines are developed, and also about vaccine safety. So we wanted to host a conversation which will also be recorded and posted to the coronavirus website on the page for folks to refer to later. The plan for today, I'm Paula Short. I am the AVP for Campus Preparedness and Response. Been working closely with all of our panelists, in fact, as we've responded to this global pandemic over the last year plus. And I'm going to introduce each of our speakers. I'm going to do their bios up front, and then I will turn it over to the experts. We will have them work through their presentations, and then we will open for question and answer as well. So as we begin, I'll introduce our panelists. Jay Evans, PhD, is Director of the Center for Translational Medicine here at the University of Montana and CEO of Inimmune Corporation. Prior to joining and co-founding Inimmune, Dr. Evans spent 16 years at GSK Vaccines, formerly Corixa Corporation, and Ribby Immunochem, working on the identification, characterization, and development of novel vaccine adjuvants, immunomodulators, and delivery systems. We also have Sophia Newcomer, PhD, from our School of Public Health. Sophia is an assistant professor of epidemiology with the university's School of Public and Community Health Sciences. Prior to joining UM's faculty in 2018, Dr. Newcomer worked for 10 years at Kaiser Permanente, Colorado's Institute for Health Research. There she contributed to national vaccine safety surveillance and research through a partnership with CDC's Vaccine Safety Data Link program. In 2017, Dr. Newcomer received the Margaret Kolczak Award for the CDC's Immunization Safety Office for outstanding performance in epidemiology and biostatistics and dedicated contribution to the Vaccine Safety Data Link. Here in Montana, Dr. Newcomer leads research focused on identifying and addressing barriers to vaccination, particularly in rural populations, and including strategies for improving public confidence in vaccines. And Dr. Jeff Adams is with us today as well. Dr. Adams received his undergraduate degree and MD from Stanford University and completed his medical residency in family medicine at the University of Washington in Seattle. Dr. Adams has been at Curry Health Center here at for 21 years and serves as the medical director for the clinic. So I want to thank all of you for spending time with us today and sharing your knowledge and expertise about vaccines. And with that, I will turn it over to the three of you.
Great, thank you, Paula, for the great introductions. I'm gonna kick it off with a little bit of background information about COVID and some of the vaccines that are available. Let me share my screen. Paula, give me the thumbs up. That on the right screen. Okay, thank you. So as mentioned by Paula, I'm a faculty member here at the University of Montana. Director of the Center for Translational Medicine. I hold a second role as president and CEO of a company right here in Missoula called Inimmune Corporation, focused on the development of vaccines and immunotherapeutics. One slide about our group. Let me turn on my laser pointer here. Well, one slide about our group here at U of M before we launch into the details on COVID. We have a large vaccine research and development team here at the University of Montana. This team was founded in early 2016 when a group of us left GlaxoSmithKline Vaccines down in Hamilton and joined the university. Since that time, the program has rapidly expanded with a lot of federal grants and contracts related to vaccine research and development. We now have a team of over 60 people here at the university focused on vaccine discovery and design. And we work on a lot of different infectious disease programs, as well as some non-traditional programs such as oncology and allergy and an opioid vaccine program, which has been in the news lately. But importantly for today, we have an active SARS-CoV-2 or COVID vaccine program that we've been working on for over a year now here at the university. So, now about vaccines, just some general overview. Vaccines are really a main driver for public health. They have been for a long time. Immunization has been called the most important public health intervention in medical history, second only to safe water, clean drinking water, and to save more lives than any other medical invention in human history. Like I said, with the exception of clean water, it saved millions of lives, and each year prevents hundreds of millions of people cases of disease. And widespread vaccination programs both prevent and control disease. As you can see, in 2016, an estimated about 86% of children worldwide under the age of 1 received the DTP, or also called the DTaP vaccine. But despite all of these fantastic advancements in years over immunization and vaccine programs, there's still about 1.5 million children that die annually from vaccine preventable diseases. So of course, this means that we can do much better. So what have we achieved so far in vaccination? Here's some examples of some great successes. Smallpox has been eradicated. Polio, worldwide disease incidence has been decreased by over 99%. Measles is largely controlled in the Americas and Europe. Of course, the DTaP vaccine has been broadly successful worldwide. What does this look like as far as the vaccines that are available? It's incredible, the developments that have happened really over the past 50 years. Anyone with children knows that there's a long list of vaccines that are available here shown on the left, and actually COVID should be added to this list as well as a vaccine preventable disease, as we're learning now. You can see things like I mentioned on the last slide, diseases like measles and polio, you don't hear about them now. And it's largely due to the efforts of vaccine researchers and the distribution of these vaccines worldwide. So now just to talk about COVID and where we are with COVID, and a little bit about the current vaccines. First off, daily confirmed case rates in the US over the past year. You can see the number, the large line there that peaked in January 2021, of course, is the US, the highest incident rate of COVID of any country, and the largest number of cases. You can see it looked pretty bleak last fall going into January, with cases peaking in January. Since then we've seen a pretty dramatic drop off in cases, largely attributed to mask wearing, social distancing, but COVID is also a seasonal virus. We've known this for years, so there's probably some seasonality as well to the drop off in case numbers as we've seen as we move through the spring. Unfortunately, within the last couple weeks, you've heard it on the news, there has been an uptick in cases again. Whether this is due strictly to the new variants or people starting to not wear masks and travel and trying to return to more life as normal is not for sure. But I think continued diligence as we distribute the vaccines is still important. How are we doing in Montana? We're following the national trend. Our peak was a little bit earlier than the national peak, happened on November 17th here in Montana. Fortunately, within the last few weeks, we haven't seen the uptick they've seen nationally in cases. Hopefully this stays this way, although I'll talk a little bit about variants here in a few minutes, and I think variants will contribute to this uptick in cases and may actually, we might start seeing the impact of that in Montana. Hopefully not, but I would expect that we might see that here as well. So now jumping into vaccines and treatments a little bit. The response to COVID has been unprecedented. As a vaccine infectious disease researcher over the past 20 years, I've never seen a response like this to a past infection of any time or a past pandemic. You can see here, we're only a year into this. And this slide shows the antivirals, they're in blue, treatments in orange, and then vaccines being developed in green. This vaccine, this chart starts in January 2020. Basically when shortly after the virus was identified in Wuhan, China. You can see, by March and April, there were already hundreds of treatments in preclinical and clinical development. You can see when the UM's vaccine program started there too, early in 2020. And this resulted in pretty quickly vaccines and treatments going from the preclinical discovery stage to the clinical stage. This chart kind of shows a timeline in history of how long it took from the first time the genetic material from a virus was sequenced to when it started human clinical trials. You can see SARS, related coronavirus in 2003, it took 20 months from the sequence to a first time in human study. And now with COVID, 3 months. And things were in phase 1 clinical trials. So I know there's been a lot of concerns raised about safety. Sophia will talk about that here in a few minutes. But one thing to rest assured is that the regulatory processes that are in place to approve a vaccine didn't change from influenza to Zika to SARS to now to COVID-19. So the vaccines that are available today for COVID would be just as safe as if it's taken 1 year versus 5 years to develop the vaccines, because they didn't compromise on safety. How are we doing on vaccine rollout? You'll notice this slide is labeled March 9, 2021, and I'll show you the updated one here in just a second. You can see vaccine distribution happened really quickly. Like I said, it's really unprecedented, the rapid with which the GMP process for manufacture of the vaccines was able to scale up to the distribution levels that they're at now. You see earlier this month, actually this was the last time I gave this summary or this talk, about 20% of Montana residents had received 1 dose, and they administered about 93 million doses through the US. You can see Montana as a state was doing pretty good compared to the others. Contrast this to where we're at today. That's just 20 days later on this chart of the vaccines in the US. Now we're over 145 million doses of vaccine that have been administered, which is pretty phenomenal if you see where we were just a year ago. About 18% of Montana residents are fully vaccinated, had received 2 doses of the mRNA vaccine or potentially a single dose of the J&J vaccine. That's 11th if you rank states across the US per 100,000 people vaccinated, per 100,000 people, the number vaccinated. So as a state, we're doing really well. If you estimate the infection rate, which varies greatly from state to state, community to community, we anticipate Montana with both the COVID positive, known COVID positive cases, and those who are asymptomatic or didn't get tested, the estimates are as high as 20 to 30% of people potentially have been exposed to COVID. You add that to the people who are fully vaccinated, and we should be approaching 50%, which is great. A lot of the experts say we need to get to 70, maybe 80% to develop what's called herd immunity and really change the numbers. As you can see, we're moving in that direction. So we're all hopeful that with more diligence, social distancing, mask wearing, getting the vaccine, that we'll get to that number here later this year and maybe be able to start opening things up. So how do these vaccines work? It's a question I get a lot. These RNA, DNA-based vaccines are new technology as far as an approved vaccine, but there are technologies that have been around for 10 or more years and are fairly well established in the vaccine community as far as preclinical and early clinical studies for safety. So type 1 is a traditional vaccine that companies like Novavax and others are developing. Novavax might be the first one that would be approved in this category. And that's a protein-based vaccine where you take a recombinant protein and add an adjuvant to it and you immunize people. This is the vaccine that people are used to seeing. Type 2 is the Johnson & Johnson vaccine as well as the AstraZeneca vaccine. It's an adenovirus-based vaccine, so it's based on a viral vector called an adenovirus to deliver the spike protein, and I'll talk a little bit more about that. And then type 3 is the mRNA vaccines, the first ones that were approved. Both Pfizer and Moderna work in this way. So this next slide here talks a little bit about what an mRNA vaccine is and how it works. So very soon after we got the sequence of the COVID-19 virus from Wuhan, China, the spike protein, which is the part of the virus that binds to the receptor on the human cells and allows it to get in. The receptor on human cells is called ACE2. So this spike protein the mRNA sequence was synthesized for that, and it's packaged in a lipid coat which protects the RNA from degradation, from RNases and other enzymes that can break it down. It also allows it to get into the cell more easily and be delivered. And so the RNA sequence is kind of a blueprint for making the spike protein. So when that lipid coating gets into a cell, that mRNA is then what they call translated into protein, creating millions of copies of the spike protein in the cell. To the cell, that looks like a foreign infection, like a virus would, making the protein a foreign protein inside the cell. The immune response can then generate an innate and then adaptive response against that, which develops your antibodies and your protective response, like a traditional vaccine. What does this look like as far as efficacy? We've all seen this. It's a 2-dose vaccine. After the first dose of the Pfizer vaccine or the Moderna vaccine, you get about 50% protection. I've seen reports up to as high as 70 or 80%. And then after the second dose, about 2 weeks, 7 days to 2 weeks after the second dose, then you're fully protected with about 95% efficacy. And this is the vaccine that most people in our community have received. We heard recently about a third vaccine being approved, the J&J vaccine or Janssen vaccine. This is based on an adenovirus vector, so it's different. It's still a nucleotide or a genetic material-based vaccine, but it's based on adenovirus. Adenoviruses have been around a long time. They can cause the common cold, fevers, coughs, sore throats, pink eye, are all diseases caused by adenovirus. But this adenovirus doesn't infect cells in the traditional sense that it's a non-replicating virus. It just gets in the cell, lets that genetic material do its thing. It's the spike protein. Make this protein so your body can have an immune response against it. Not so dissimilar to the mRNA vaccine. It's just a different way of delivering that genetic material so that your body, your own cells of your body can produce that protein and your immune response can respond to it. And so the difference between this is it's a little bit more stable as far as a vaccine vector. So it's stable at 2 to 8 degrees refrigerator temperature, which is critical, especially when you're trying to distribute vaccine worldwide to third world countries where you don't have -80 freezers to store an mRNA-based vaccine. So this is going to be critical for not only vaccination here in the US, but also worldwide because the storage, it's not quite as effective as the mRNA vaccines. You can see 70 to 85% effectiveness. But the thing we don't know yet, because these vaccines are new, is what's the durability of the response. We don't know if the mRNA versus the adenovirus vaccine is going to have a longer lasting durable immunity. So it's hard to predict which one is better to get. And what I tell people, you get whichever one is available. If I went in and the J&J vaccine was available, I would get it. As it turns out, at the Curry Health Center, the Pfizer vaccine is what was available. I got my second dose last Monday, and I'm happy about that. And I encourage all friends and family, as soon as they're eligible under the guidelines to get the vaccine, to go get it. Because as you'll hear from here in a few minutes, the safety profile of these vaccines is excellent, especially when you go against the risks of getting COVID and some of the outcomes of that. So, a big topic right now is the variants. You hear it almost every night on the news. What's going on with these variants? How are they working? Here's a map of a recent map of the variants where they've been detected in the U.S. You can see Montana's shaded green. We don't have the number of cases of variants in Montana and some of the other states, but I think you'll rapidly see these increase across the U.S. because of their increased transmissibility. What does this look like as far as the different types of variants circulating in the U.S.? Most of the variants that have been identified worldwide, most of the major ones, are now in the U.S. The most common one is the UK variant, but the Brazil, South African, and there's also a homegrown variant, I call it, from either the California variant or the New York variant that are also circulating. But by far the most common one is the UK variant. One thing to note, there's not a lot of information yet on these variants, and we'll talk a little bit about how the vaccine works with these variants. But here, one notable thing is this one mutation that's common to the UK, Brazil, and South African variants, this N501Y mutation, a single amino acid mutation. when they took neutralizing antibodies from convalescent serum, those are people that were infected with the current strain of COVID. 48% of those were not very effective in neutralizing this new variant. And while there's not a lot of data out there, what this tells to me is that even people who tested positive for COVID over the last year should be getting vaccinated. Because it appears as though the vaccine might actually provide better protection against these variants than being infected, which from an immunologist, I don't understand why that would be, but some of the early data suggests that might be the case. So even if you've had COVID, you should consider getting the vaccine to make sure you have the optimal immunity as these variants start to circulate more broadly. And now the big question that everybody's asking right now, and the data is really early and there's not a lot of data to support it, but what is out there is summarized here. Are the current vaccines effective against these COVID variants? Boxed here in red is the effectiveness against the current vaccines against these variants. You can see it showing 90 to 100% effectiveness at preventing variants in these populations, which is really exciting, great data. This is what we all want to see. But the numbers are really, really low. We're talking, for the Moderna vaccine, only 185 people have come, have been in the control group versus the vaccinated group have had one of these variants. So the numbers are really low. So we're still getting more and more numbers, or building on this every day. If you take serum from people who have been vaccinated and you do an in vitro, that is in the lab-based neutralization test, and how much serum does it take to neutralize these variants? I think this gives us a better indication of maybe 6 months from now where we'll be as people's antibodies titers, start to change over time after vaccination. What you can see is that it takes more serum to neutralize some of these variants in these vaccinated people. For the Pfizer vaccine, it takes between 2 and 6 times more of the neutralizing serum to get the same neutralizing response, which takes a higher antibody titer, which tells me that over time, as people's antibodies start to drop off after vaccination, which always happens, the first thing you're going to be susceptible to is one of these variants, which is why we need to stay on top of vaccination. So my hypothesis, there's no evidence supporting this yet, is that over time, as your immunity drops off, we're probably going to need a booster vaccine. And that booster vaccine will probably be made up of some of these variants. For example, this N501Y mutation would be an obvious mutation to include in a follow-up booster vaccine, which you might need to get a year from now, maybe 3 years from now, as we learn more about this. And the great thing about these mRNA and adenovirus-based vaccine technologies and how much we've ramped up production is that to develop that new booster vaccine is going to be very, very fast. All they have to do is change the mRNA sequence, and they have a safe vaccine ready to distribute. They have just the distribution channels through the public health departments already established. And so that is all I had to present today on the kind of the current vaccines and where we're at in the efficacy and where it's at with the variants. So I will hand it over to Sophia.
Paula, are you seeing my slides? Okay, and you're seeing my slides, not the notes. Yeah, and volume good? All right, great. Thanks, Dr. Evans. So now that we've learned about how these vaccines work, I'm going to talk for a little bit about how we know that they're safe. And so I'm going to start by talking about what my definition of vaccine safety is. So when we talk about safety, we're not talking about the absence of risk. No medical intervention is 100% safe. So to me, I think of vaccine safety as a field where we seek to study, quantify, and communicate about the risk of vaccination. And we do this so that individuals and societies can make risk-benefit decisions about vaccines. And really, the central challenge to vaccine safety researchers is figuring out whether health outcomes occur more frequently following vaccination. So if we think about it, there's health outcomes occurring at a baseline rate, and we seek to look at whether there is an uptick following vaccination. And if we do find this, then we call it an adverse event from the immunization. So in the US, we have really high standards for the safety of vaccines for a few good reasons. First is vaccines are a medical intervention that we give to healthy individuals. And as such, we have less of an appetite for adverse events than we do for drugs and for therapeutics that are given to sick people to help make them better. Another reason is that the scale of this medical and public health intervention is huge. Vaccines are recommended to everybody at different phases of your life. And right now, we want all adults in the US to get the COVID-19 vaccine. So to be successful at such a big scale, vaccines have to be safe, they have to be effective, and they have to be accepted by the population. And finally, vaccines are our strategy in the US for infectious disease control and prevention. You saw from Dr. Evans' slides, as vaccine rates have gone up for certain diseases like measles, rates have declined. Our lives would look a lot different if we didn't have vaccination. So we really can't afford to lose public support in this strategy for infectious disease control. And so as a result, the United States invests substantial money, time, and person power to making sure that vaccines are safe. So I'm going to talk about how we do that in the US. So here is a figure of the vaccine life cycle. And I'm not going to go through in detail, but basically, if we start over on the left, vaccines start in laboratories. Right here at the University of Montana is one example, where chemists and immunologists are working to discover antigens and adjuvants that might be potentially successful in vaccines. And safety of these antigens and adjuvants in humans is top of mind during that discovery phase. Once it moves out of the basic science phase and it's ready for human trials, it moves into this green phase in the middle, where vaccines are tested in humans for their safety, their effectiveness. And we're going to talk today about phase 3 trials, which is what we've heard a lot about in the news. These big trials that were done by Pfizer, Moderna, and Johnson & Johnson, and other companies are doing trials now to test the safety and effectiveness of these vaccines. Finally, once phase 3 clinical trial data is available, the FDA is charged with authorizing or approving use of vaccines. It then gets passed on to the CDC, that's charged with recommending use of the vaccine in the population. And then finally, we have this purple area over on the right, phase 4. So vaccines continue to be studied once they're being used in the population for years and even decades. And so today I'm going to share data on both what we've learned about the safety of the COVID-19 vaccines from this clinical trials phase here, but then also what we're learning about the safety of vaccines now that we have many millions of doses used. Before I jump into the data, I just want to share that this is not all, there's many eyes on this process before a vaccine is recommended for use on the population, including independent groups of experts. So it's not just pharmaceutical companies and the government making decisions here, but at least 3 key phases, we have groups of independent experts with their eyes on the safety data. So that happens in the clinical trials during the phase 3 process. There's a data safety monitoring board that reviews the safety data and is communicating to FDA as to their analysis of the vaccine safety. The FDA has a group of advisors called VRBPAC. This is a good alphabet soup of acronyms here, but VRBPAC is the Vaccines and Related Biologics Product Advisory Committee. And then finally, there's a group of independent experts called the ACIP, the Advisory Committee on Immunization Practices. They also review the safety data before making recommendations to CDC about who should get the vaccine in the population. So these are groups of physicians, scientists, many people at universities, again, independent of government or pharmaceutical companies looking at the safety data. So now let's dive in and talk about what we've learned from clinical trials about the safety of vaccines. of these COVID-19 vaccines. So one thing we've learned is that there are non-serious adverse events following vaccination that are quite common. So these fall into two broad categories. We have local reactions, which tend to be at the injection site. So your arm might hurt, redness, swelling, and then also systemic reactions, which are like fever, feeling tired, body aches, etc. And the prevalence of vaccine recipients reporting these common non-serious adverse events was pretty high. The majority of recipients did report local or systemic reactions. And this was more common in the 2-dose vaccines following the second dose, and also more common in the younger participants than in the older participants. So this is one thing we learned from clinical trials. Now, compared to other vaccines, the prevalence of local and systemic reactions are a bit higher than what we see, for example, with flu vaccines. But again, these are non-serious. But this is just to kind of let people know ahead of time, hey, you might feel a little bit crummy after getting your vaccine. I got my first dose of Pfizer last week, and the next day I kind of had some muscle aches and I just wanted to rest for a few hours. To me, totally worth it, because I know that I'm building my protection and my immunity against a very dangerous disease. Let's talk about serious adverse events. The good news is there were no major safety concerns identified in large phase 3 clinical trials. So one thing I want to point out here is, first of all, how large these phase 3 clinical trials were. So when these trials were getting tested, they were tested in anywhere between 30 and 43,000 individuals, depending on the trial. These are quite large. And this was one way that these vaccines were able to come to market so quickly, is that the pharmaceutical companies really committed to doing very large trials in a short period of time. And we had a lot of Americans who are willing to raise their hand and say, yeah, I want to be part of this effort. So I'm really grateful to the many volunteers who participated in these trials. Serious adverse events are defined as adverse events following vaccination that could lead to hospitalization, disability, or death. And if you look here between the vaccine group and the placebo group, so remember in these trials, about half the people got the vaccine and half the people thought they might have got the vaccine, but it was really a placebo. The percent was pretty much equal and very low in terms of serious adverse events following vaccination. And then FDA reviews every single serious adverse events reported in clinical trials, and they found that only a handful might have possibly been linked to the vaccine. This included things like shoulder injury. There were also 2 reports of facial swelling in persons with a previous history of cosmetic filler injections. A small variety here, all of which were self-limited and did resolve. So overall, taken together, this is very good data regarding the safety of the vaccines that came from the clinical trials of these COVID-19 vaccines. So now I'm going to turn to talking about, now that the vaccines have been out for a few months and we have many millions of individuals who received them, what have we learned, continued to learn about their safety? So first of all, I'll point out that there are at least 10 different systems and thousands of individuals working to continually monitor the safety of vaccines now that they're being used in the population. So this is a diagram I took from the CDC, and we'd probably need 4 hours if we were going to walk through every single system at work. But I'm just going to talk about 2 of the systems today. I'm going to talk about the VAERS system, the Vaccine Adverse Event Reporting System, and then VSD, which is what I used to work for, which is the Vaccine Safety Data Link. And I chose these 2 because they're 2 different types of surveillance systems, and they're also systems that have been around for decades. So these systems have monitored the safety of many previous vaccines before this, and they were certainly well equipped and ready to study the safety of COVID-19 vaccines. So VAERS is often called our early warning system. So VAERS is co-managed by the CDC and the FDA. And basically the way it works is it collects reports from anyone about potential adverse events following vaccination. So anyone can go online, or you can even fill out a paper form and send it to CDC, but you can go online and you could report what you think might be an adverse event following vaccination. And medical providers are required by law to report certain adverse events following vaccination. So the way it works is CDC CDC analysts are working to look at the data coming in, and then they're comparing what they see to what we expect in terms of baseline rates for certain adverse events. And I'm going to share in a minute some early reporting from CDC in terms of what they're learning from VAERS. But first, I want to point out that VAERS information can often get misinterpreted, and I see this sometimes in social media and on the internet. So there's a lot of examples of how I've seen VAERS misused, either in traditional or social media. First of all, I've definitely seen some scare tactics out there where people will pull out a VAERS report and say like, oh, there's been a report of death following COVID-19 vaccination. You also can't just look at VAERS numbers on their own, right? Like you don't really know, okay, if I've seen this many reports of an adverse event to VAERS, is that what I'd expect in the population anyway, even if vaccination weren't happening? Or is it due to vaccine? Or is it perhaps due to the vaccine? And so this is just a reminder to really be critical about online sources of vaccine information. And I include this image here, which is actually from the late 1800s. And this is just a reminder that as long as we've had vaccines, we've also had people trying to scare other people about vaccines. And this was from an anti-vaccination pamphlet that was being circulated in the late 1800s. And, you know, I'm sure if you are ever online and you're looking up information about vaccines, you might find some memes or different images that are also quite scary. And I just want to point out in terms of online sources of vaccine information, there was actually a study done in 2018 by computer scientists that analyzed the sources of anti-vaccine information online. And they found that a good chunk of anti-vaccine information online in the US actually came from foreign sources, including Russian bots and trolls. And so just like we have foreign entities trying to sow political discord in the US, we also have foreign entities trying to influence our vaccine decisions in the US. So here I just put some reputable sources of vaccine information, including the CDC, which I think is the best source. But going back to VAERS, which is a valuable source of data, I'm going to just summarize what the CDC has reported out in terms of what they learned from VAERS after the first month of the COVID-19 vaccine rollout. So during that month, almost 14,000 vaccine doses were administered. There were about 7,000 reports to VAERS. In terms of deaths, there were 113 reports to VAERS, the majority of which were from long-term care facility residents. So as we know, people in long-term care facilities tend to be of advanced age. They tend to have many medical comorbidities and are at higher risk of death. And so based on data that we have, the CDC estimated that among the 1 million long-term care facility residents vaccinated during that time, we'd actually expect 7,000 deaths. So certainly VAERS wasn't giving us a signal of an increased risk of death following vaccination. And in fact, additional research has since been done comparing mortality rates among vaccinated and unvaccinated long-term care facility residents. It seems like vaccinated residents have lower mortality rates, likely due to protection from COVID-19. Finally, anaphylaxis, which is a severe allergic reaction that can happen after the vaccine. And there were definitely media reports when the vaccine first came out about people having this immediate severe reaction. Well, anaphylaxis is a known, very rare adverse event that can happen after vaccination, particularly among people who have a history of severe allergic reactions. So based on VAERS data, it's now estimated that we expect about 4.5 cases per 1 million doses. And this is actually in line with what we know about other vaccines. And this is going to be the reason that when you get your vaccine, you're going to be asked to wait for 15 minutes. Because typically, if you are going to have a very rare adverse event, again, very rare, it's going to happen immediately. And all immunization sites are ready to treat a severe allergic reaction. Finally, I'll wrap up by talking about the another large data source, which does surveillance in a different way. This is the Vaccine Safety Data Link. The Vaccine Safety Data Link, VSD, is a network of health plans around the country. They provide care for over 11 million individuals in the US. And what they do for vaccine safety surveillance is they pool all their data together. So they have these electronic medical records on who's getting vaccinated, and then what's happening to them. Are they coming to the hospital? Do they have certain diagnoses later? And we're able to use all that information to conduct large-scale vaccine safety surveillance. And they're doing that right now with the COVID-19 vaccines in near real time through the VSD Rapid Cycle Analysis Program. And I'm not going to go through this table in detail, but basically they're looking at a whole bunch of outcomes, including anaphylaxis, including Bell's palsy, which is a facial paralysis, down to blood clots. And they're looking to see whether people are having more of this event following vaccination as compared to people who aren't vaccinated yet. And as of when this data was released, there were no statistically significant increased risk. And I think the data continue to be monitored with no major safety concerns identified. So to wrap up, as I discussed, we have really high standards in the US, and we do a lot of work to make sure that vaccines are safe, and we continue to monitor their safety. All the data we have about COVID-19 vaccines are really reassuring. And we have to accept there's some uncertainty here. We can't talk about really long-term safety because this vaccine hasn't been available for years. And also, it is possible that once the vaccine is being given to large groups of people, we might detect very rare adverse events. But again, we've already given the vaccine on a pretty large scale, and all the data we have right now is really reassuring. And we also have to talk about the cost, the risk benefit, right? There is a human cost to delaying vaccine in terms of disease spread and death from COVID-19. So I'm certainly convinced that the data that we have supports the safety of the vaccine and supports its effectiveness in fighting this pandemic. So with that, I'm going to turn it over to Jeff Adams to talk a little bit about the clinical perspective here.
So thanks, Sophia. So first of all, thank you, because we've worked together for so long trying to make sure we had that campus safety plan. But the one thing I want to say is that I want to wrap up early so that we can actually take questions. So I'm going to make this a 2-minute long presentation. The first thing I want to tell you is that we did about 4,000, a little bit more than 4,000 COVID tests over the last year here at Curry Health Center alone. Those are the number of people we saw that were worried they caught COVID, and the implications of that were being out of, quarantined, isolated from their families. But I also want to tell a story of people that didn't die from COVID, but had disability from COVID. We know that 134 people from Montana died from COVID, but they didn't list the people that had injury from COVID. We saw athletes that had some cardiac damage from COVID that had to be out of athletics for a while. We saw people that had lasting fatigue, myalgias, headaches from COVID infection that have still gone on to this day. I know somebody that is in her 20s and she's still having that fatigue problem, mental cloudiness. And that's concerning. I mean, that we don't know the end of that one. We also have people that have lost their taste and smell from this virus. So I want to tell people that just because it doesn't kill you doesn't mean it doesn't hurt you. We have done almost 11.5 thousand vaccinations here out of our health service with our health service team. So we have a lot of experience doing this, and we don't have anything that was major. We had some minor reactions. We certainly had an awful lot of people with sore arms, people that felt a little fluey for a day, people that felt tired that just had to go to bed early. But all those people rolled through that and are out the other side now feeling protected. I'm vaccinated now. My wife's vaccinated. Anyone that I talk to, I try to get to be vaccinated. It's a protection from us from death from COVID, but also injury from COVID. And it's also a release from the life that we've had to live over the last year that has been very confined and really devoid of the pleasures that we usually have week to week, whether it's with friends or family or having events together or just going out and seeing music. So I would do it. I would recommend it to anyone I know, but I would tell you one thing. If someone's resistant to vaccination, be kind and patient with them. It doesn't really work to just argue against them and tell them that their views are outdated. It's better to try to understand them and then let it sink in for a while and just let them sit with the idea that maybe consider vaccination. If you could consider it, could you consider a date that maybe it would be the right date for you to start participating with this vaccination program? We're here to answer all questions, but let's wrap up so we can hear questions from our audience.
All right. Thank you again for a very informative presentation, 3 of them in fact, and we will open to questions. So if folks have questions, you can hit the Q&A. I will be reading those to the panelists and they can pick who would like to answer or if all 3 would like to respond. And I had a couple of questions of my own as well, but first we'll go to our attendees. A question, do you have any specific comments on pregnant women receiving the vaccine?
That is a really good question. I have a very good friend of mine who is pregnant and had to recently navigate this decision with her healthcare provider. And so I'll say a few things. One is that we do know that pregnant women were not included in clinical trials, and that is pretty common, although some clinical trials participants did become pregnant during the trial. And so there isn't a ton of data from the clinical trials, but there is data that basic scientists have looked at regarding how the vaccine might affect the mother and placenta. And so I think right now, the guidance from the Advisory Committee on Immunization Practices is that pregnant women can get vaccinated and they are welcome to do so. And that should be a decision that is made with their medical provider. In the case of my friend, if anyone wants a personal example, she did choose to get vaccinated after having that discussion.
Yeah, we've had many pregnant women get vaccinated through our vaccination program here. I don't know of any problems with that. And I feel like they have a protection against for their newborn as well coming out, because they'll get some maternal antibodies that we passed along. So not studied, but probably safe and probably no additional hazards.
Yeah, and I will say there's a lot of post-marketing studies. So now that we do have pregnant women getting vaccinated. So one system that I know is working that I didn't get to talk about is this V-Safe system, which was set up by the CDC. And when you get vaccinated, you might see a sign or you might get a piece of paper where you're invited to, where you can actually participate in V-Safe, where the CDC will actually send alerts to your phone and ask you to report adverse events. So they were specifically looking at enrolling pregnant women so that they could closely study any adverse events in that population.
Great. Next question. What are your responses to the vaccine being called an experimental biological agent for a 2-year study? I would dovetail onto that I've also heard that, if you get the vaccine that you may or may not get the vaccine because you're still in a trial, which I know is not true, but I wanted to just mention that to you and see your response for that as well.
I actually haven't heard that term, experimental biological agent, being used to reference people getting the vaccine. Maybe, Jennifer Sevilla have heard that or discussed as far as within the medical community. I would say that this is an emergency use authorization through the FDA. It's not the full BLA, Biological License Agreement authorization through the FDA, which will come in the future, which in some groups might be considered an experimental because the emergency use authorization is all that I can think of. And biological agent, a lot of vaccines are biological in nature. In other words, they're purified from a source that's a biological nature. The mRNA vaccine is a fully synthetic vaccine in the lipid coating with an mRNA that was made synthetically. So it's not even necessarily a biological agent. The adenovirus vaccine, of course, is a cell culture derived based vaccine. So that would be considered a biological agent as far as a vaccine. Any comment from the other two on the experimental biological agent?
Yeah, I mean, it's certainly not experimental anymore because the experiments have happened. Those are the large phase 3 clinical trials where we learned about the safety and efficacy of the vaccines. Following along to Dr. Evans' point, these vaccines are approved through an emergency use authorization, which is unique. We haven't really had that before for vaccines, but we also haven't really had a pandemic of this scale either. But really, the only difference between emergency use authorization for these vaccines versus the standard approval, which Pfizer announced they're going to apply for this month, is the how long you follow a participant for. So the size of these studies were big, the data they collected, the thoroughness of the methodology, all of that was very standard. And in fact, the size of these trials were bigger than what we sometimes see for vaccine phase 3 clinical trials. The only difference is that we wanted these vaccines to get out as quickly as we could after we knew they were safe. So they had 2 months of safety data. And once they have 6 months, they're going to be applying for these the standard licensure.
Okay. Again, if you have questions, go ahead and put them into the Q&A. I had another question, and it was related to the response post-vaccine. So if you're somebody who didn't have any like you felt fine, you didn't really have any sort of reaction, does that mean that you didn't have a good immune response, and you're somehow not as protected as someone who actually got kind of sick and then got better in terms of, you know, antibodies and how with their level of protection?
That is a great question. I have not yet read a publication that matched their mild, you know, local adverse events that happen following vaccination with the titer. I expect we will see that come out in future years, a relationship there. You know, I know we're having on campus, we're having It Starts With Me thing happen. I think it's happening this week. Tomorrow morning, I go in for that. And one of the tests I'm getting done is serology. I want to, you know, I'm curious as to what my COVID-19 antibody levels are. More of a personal curiosity. There's no chart that I can plot it on to say I'm protected or not protected. That data will come with time. There's a lot of work yet to be done on the durability of the response. As we know, some vaccines last, very few, but there's a couple out there that seem to provide lifelong protection. Others, you know, immunity goes down over time. You have to have, you know, a booster tetanus vaccine after so many years. It's pretty common that antibody responses go down over time. So where do we draw that line in the sand for COVID, where people will need a booster, or maybe people that received 2 vaccines that didn't respond as well will need a booster? The community is still determining where that line is in the sand and what those antibody titers are that correlate with protection against the circulating strain. So I think time will tell.
Okay. Another question, is there any research on whether the vaccine could affect women who are not currently pregnant but are planning to have children in the future? Is there anything of concern there?
Yeah, I certainly don't think there's anything of concern. I mean, like I had mentioned, there were people in clinical trials who became pregnant. And as we saw, there were no serious adverse events. Again, that's a small group. I think that's a topic that will continue to be studied. But I would also just invite thinking about the kind of opposite about not getting vaccinated. So we do know that if you're a young woman who's looking, you know, of childbearing age, looking to have children, we certainly know that COVID-19 or getting infected with that could certainly derail those plans. So, you know, absence of any evidence and all the evidence that we do have supporting the safety of vaccines, certainly we know that these vaccines are safe among women of childbearing age.
For me, I've had this question a number of times over the past few months, and my answer has always been there's not a lot of data. But to me, as an immunologist, the risk of either someone who's currently pregnant or planning to get pregnant of getting COVID versus the vaccine, the risk that you have with COVID and the long haulers and the stuff that Jeff talked about, these long-term effects that we're still learning about, those risks are much higher than any risk associated with the vaccine itself, as of the data that's out today. So that's what I tell people. It's a personal choice because there's not a lot of data, but the data supports that it's much lower risk to those individuals getting the vaccine versus getting COVID, given the circulation rates of COVID in the community today.
Yeah, I saw the same in terms of the CDC review showing no additional risk for women to cause infertility. But I also would echo what Jay said, that women that become pregnant now enter a higher risk category for COVID complications. So it's just something to think about.
Let's talk a little bit about AstraZeneca. I know that that's not a vaccine that's currently approved by the FDA under an EUA for the United States, but obviously making news abroad. The specific question is, do you expect AstraZeneca will be approved in the US? And do you have any insights on the safety of this vaccine?
I don't. I follow it. I have a friend and colleague who's on the VRBPAC. Of course, he can't provide me information. That's all private. So I know there's a lot of concern. There's some concerns about blood clotting in some places. I just saw, I think today or yesterday, that Canada was putting a hold on it because of, you know, potentially up to 1 in 100,000 people might have an issue with the blood clotting disorder disease. But other reports are 1 in a million, or maybe background. So I know there's a lot of stuff in the literature. Again, I recommend people go to the CDC site, the FDA site. I haven't looked to see if the FDA panel has released their findings. safety versus efficacy on that vaccine yet. So that's where I would say continue to monitor the CDC and the FDA. Those are the people who are experts on vaccine safety. And if it becomes approved through the FDA, through EUA, I'm assuming that the FDA would not approve a vaccine that's less safe than the ones that are currently approved. If anything, the bar is even higher now because we have approved vaccines that are effective. They're not going to put out something that's not as safe as a vaccine that we currently have.
Okay. Another question specific to you, Jay. What is the status of the vaccine work that you're doing or leading here on campus?
That's a great question. I didn't present those slides. So we have an active vaccine research program for COVID. It's been ongoing since February. Ours was never designed to be the first to the clinic, to try to beat the mRNA or adenovirus vaccine technology at the clinic. We are already working on vaccines for tuberculosis, improving the vaccination for pertussis, universal flu vaccine. What we do is drive immune responses that are lifelong durable and broader in protection. We design vaccines that, for example, in the case of COVID, we're doing this with flu where we're looking at how broadly against the different flu variants that we can add immunostimulants to drive a broader protection so you don't have to get a seasonal vaccine. Maybe we have a super seasonal vaccine. So we're doing the same with COVID, looking at if we can broaden that immune response so the vaccine is protected against not only the current variants, but future variants. So we're looking at that next generation vaccine that'll provide that broader, more durable response. So maybe people only have to get vaccinated, every 10 years or maybe once in their lifetime. And so we provide all that information back to NIH, and then they can choose which of those vaccine candidates that we're testing here at the University of Montana, which isn't only ours, or vaccines from other companies, could potentially move forward to the next generation vaccine in clinical trials.
Okay, next question. Are any of you aware of widespread serology testing plans to ascertain immunity overall? I know you talked a little bit about herd immunity, but I guess that would be one way to try to get at that more precisely.
So I know that the companies that are distributing these vaccines are very interested in that question about durability of the vaccine, how broadly they're protective against these variants. You saw some of the data that I provided. I don't know of any widespread government, CDC initiated plans to look at that. I think most of them are driven through the NIH, academic investigators and companies trying to understand where the lines in the sand can be drawn for protection against the circulating strains and variants. I don't know if Jeff or Sophia, if you've heard any CDC or FDA driven serology that's ongoing on that scale.
Yeah, I'm not sure. I mean, there's two things when we're talking about the effectiveness of the vaccine. We can talk about serology and we can talk about antibody response, but then I think CDC is also very interested in the clinical outcomes there. Is the vaccine protecting against clinical COVID-19? And so I do know that just yesterday, a CDC-driven study that involved multiple health plans around the country, they released their data on real-world effectiveness of the vaccine, showing that the vaccine had 90%, the mRNA vaccines had 90% effectiveness in preventing clinical COVID-19 disease.
Yeah. There's publications coming out almost daily on these types of questions. But that's a great question. I mean, now that we have all of these community health centers, counties and at the state level geared towards COVID vaccination, as that becomes, it's broadly available now, but as we approach herd immunity, it would be great to turn some of those nationalized resources that are in place towards serology testing and understanding the durability of that response. I think it's a great idea. I haven't heard of that in the works.
Okay. Those are all the questions that had been submitted, and we're just about at time. I guess I would just ask one final question for all of you, and it's related to a common concern I'm sure you've heard as well, and we talked a little bit about it, and that is this overarching concern about the speed with which vaccines have been developed. And I think sometimes people sort of think about that and think that maybe there was some fast tracking and therefore maybe some corners cut, and just what could you maybe even arm us with to say when someone says, oh gosh, I'm really scared about this because it just went too fast? Well, maybe we'll close with that if you guys have any feedback.
I guess my brief comment on that is that being in the vaccine industry, hearing from a lot of our collaborators and other companies whose clinical trials were shut down, to give the FDA the time to rapidly respond to the COVID vaccine questions. Submitting phase 2 and phase 3 clinical trial plans before your phase 1 clinical trials were finished is unheard of. And so that they didn't compromise on safety, they simply cut the red tape in the system needed to get a vaccine through faster without compromising on safety. That's my take in a nutshell on how they were able to do it so fast.
Yeah, I mean, I agree. I point to 3 key reasons. One is the removal of red tape that happened that made things go faster. 2, which is what Dr. Evans talked about, is that this technology, the mRNA technology, was already developed. It had been under development for 10 years. So it wasn't new, even though it hadn't previously been used in vaccines that we currently use in the US. And then 3rd, which I pointed out, is these companies went really big with their trials, and size matters when we talk about vaccine safety. You know, the more people that we have in a study, the more likely we are to detect something rare happening. And so they went really big with their trials and were able to enroll a lot of people in a really quick time frame.
Okay. All right, well, we are at time. I wanted to say thank you again to our panelists and to our attendees as well. As soon as it's available, a recording of today's webinar will be available on our COVID website, so folks can check there. And thank you again, panelists, and thanks attendees for taking some time to have this important conversation about COVID vaccines.